You know alcohol? You know alcohol?
Nope.
Oh.
He's got an attention device. If you could wait 20 seconds, you can get
Got my
that.
attention.
All right, morning everyone. So you all probably know what I mean when I say evolution, but if I say something like comparing punctuated equilibrium or phyletic gradualism, probably lose half of you. So today I'd like to explain evolution by going over the Michigan State Long-Term Evolution Experiment. It's an ongoing experiment. It was started by Dr. Richard Lenski at Michigan State University. And you guys might not get excited hearing about evolution as much as I do talking about it, but you should, because it's really fascinating and evolution's everywhere. It's really amazing. So I'll go over the setup of the experiment, some of the amazing things they observed, and then what that could mean, what some of those observations could mean. The experiment was carefully planned and carried out. Started in 1988 in February with a single E. coli bacteria. What they did is they took that bacteria, and those things break down into two bacteria and they have the same DNA. And so they were able to fill a flask that had glucose in a standard solution. And then they took like 1/10 of a milliliter and put it into each of these 12 flasks, so they all have the same DNA. And then they could every day take 1/10 of a milliliter into a new flask with a new standard solution for each of the 12 lines, and those 12 lines would all evolve independently. The only difference in mutation or in DNA would arise in mutations that can then overtake in the population and cause changes. They could also freeze older generations and bring them right back to life, and do really crazy comparisons and stuff like that. The one difference they actually did have in DNA, though, is an ARA marker that in 6 of them they switched on so they could plate it and it would be pink compared to the other being white. And they could do really cool stuff with this, not only plating them like this, but taking 2, putting them in the same flask with glucose and letting them grow, and then comparing that, seeing what color it is. It also helped them with cross-contamination. When they did their daily turnover and took one flask to the next, they would go a plus and then a minus and then a plus. So if there was any contamination, it would show up as pink in the flasks. There are a lot of... this was carried on for years, and they took data on cell size, metabolism, and growth and other things. So gradual evolution took place. This is a picture after 10,000 generations of bacteria growth. These are the 12 different lines. They all started at the same exact size, and they all eventually grew a little more. Because in this solution, in the flask, it wasn't like nature, and so they evolved little things to allocate the glucose and whatever in the solution better. And increased cell size obviously helped doing that. There's a lot of different things that contributed to cell size and the change there. And 2 of these lines are actually pretty close to the same. And it was for the ARA1 and the ARA-1. And so they took the genes and actually looked at them for these 2 different strains. And there were 59 different point mutations in the ARA plus 1 that they compared to the initial bacteria. And when they looked at the ARA minus 1, there was still the same exact 59 changes, which in a random experiment would absolutely never happen. You never have the same exact 59 out of billions and billions of changes that could occur, happen in 2 different things out of 12. But with natural selection, it actually makes great sense because they had the same conditions, and so the same mutations occurred and brought on the same changes in the cells for that environment. So that's all gradual evolution. It's really cool. But around 30,000 generations, this happened. Now, it might not look that cool, but this is one of the coolest things that we've ever observed in evolution ever. And this cloudy flask right here is cloudy because there's actually a lot more bacteria in it than in any of the other flasks, and more than there was in previous generations. And it's not because they put more glucose in or they put more bacteria in. is because the bacteria actually evolved the ability to use citrate that was in the standard solution to break down glucose even more. And so it could pull 18 times the energy from the glucose out now, because basically it could breathe like humans can, bring in oxygen, breathe out CO2, and it would just attach the carbons to the glucose, and it could pull energy from all of it. Really cool stuff. And so they were thinking, how could this have even happened? Something like this, like E. coli, one of their defining things is that they can't do aerobic respiration. They can't use oxygen to break glucose down more. But these were. And so they looked back at previous generations of this same line, and they could actually get the previous generations to re-evolve this by just putting them in their own flask line and letting it go. And then by doing that and checking earlier and earlier genes and stuff in the same lineage, they figured it out. There was actually several point mutations that occurred that each individually had absolutely no effect on the fitness or survivability of any of the bacteria. But with all of them together, they actually allowed the chemical reactions to take place in the bacteria to where it could do this. And all it really takes is one bacteria in the entire solution to have all of these things, and it'll be able to reproduce faster and overtake the flask faster, and then every subsequent generation, that bacteria would take over and it'd be present. So that shows an irreducible jump in complexity, which is just awesome. So what does all that mean? Well, the whole experiment kind of makes a great case for natural selection as a whole, because it shows the subsequent generations being more adapted to survival and more fit than the previous generations were. But remember when I mentioned gradualism and punctuated evolution in the beginning, it actually makes a great case for both of those, because the gradual evolution, you can see with like cell size and other changes, they're really slow, and it's just one little mutation can affect maybe the size of the cell or something, and that just takes over in the population. But it also shows punctuated equilibrium, where you might be at a stasis for a while, and then a huge change causes the entire population to change, and then it might sit a while and then change again in a really big way. So the whole experiment is just awesome. Pretty much is our best showcase for natural selection in a lab. I hope you guys enjoyed hearing about it as much as I enjoyed talking about it. Thanks.
What's your major again?
Computer science.
Right.
But I freaking love biology.
All right, so Michael, what did you think?
I like the topic a lot.
I didn't know if you're okay.
Seeing the different blasts and stuff as a beginning, that really made you wonder what he was going to talk about. He also made a lot of really good points, like when he was saying how the two strands have the same amount of mutations, even though there's billions of different kinds. Just thought of that happening in a randomly or just would never happen, but shows how like natural selection kind of manipulated them into evolving along the same path. But I thought it was really informative and well done.
All right, Philip, there's a lot of stuff in here that I like. I think you did a nice job of kind of identifying what the topic was, having an interesting way to get started, and giving us a good justification for it. You have a preview of the body of the speech, but you do kind of rush it, and you drop your voice while you're speaking about it, which I think makes it a little bit harder to follow the organization during the body of the speech. The one thing that helps recover that is that you've got good slides for each of the sections that you're going to, and it's the movement between the points seems very naturalistic, so it does feel like it is developing appropriately. I do think that you need to have a little bit more citation of the material from the researchers themselves and from others who have commented on this particular experiment about what it means. I also think that you sometimes use terminology that probably is understood by people in those sciences but may not be understood by others. And frankly, at the end when you're talking about gradual and punctuated equilibrium and evolution, I'm going, okay, now what does that represent and how does that work? And you gave a little bit of an explanation right at the end, but I didn't think it was